All biological organisms age, and progressively become older and older. Well, almost all. For those of us who don't have the luxury of reverting to a polyp, we are subject to a finite lifespan, and as Father Time marches on, an accumulation of changes occur as we age. For us humans, some of these changes are obvious, like when our skin starts to wrinkle or our hair starts to gray. Other changes, like our body's overall efficiency, are not so easy to visibly identify. But why do these changes happen? Is aging fundamentally unavoidable? Well, as expected, the time between our conception and passing is a complex process that's still not fully understood. But let's start simple. What do we mean by age anyways? Well, to start, all organisms have a chronological age and a biological age. Our chronological age is simply a number that quantifies the amount of time you've been alive. On the other hand, your biological age is relative functional efficiency and capability of your body at that age, which varies based on a multitude of factors. To distinguish the two, consider the following. John is 50 years old, but has a mental functioning and brain plasticity of a 20-year-old. While a trip around the sun on Earth is 1 year to me as it is to you, it does not mean we necessarily change at the same rate. Of course, this begs the paradoxical seeming question. Can we live more years without aging? While there aren't any immortal humans yet, one area of research related to lifespan are telomeres. Now, what are telomeres? Coined by Hermann J. Müller in 1930, telomere comes from the Greek word telos, meaning end, and meros, meaning part. These cap-like structures exist at the end of linear chromosomes, and consist of non-coding nucleotide repeats. While not universal, the TTA-GGG telomere repeat sequence is present across all vertebrae, showing a high level of conservation. Think of telomeres like the aglets on the ends of shoelaces. Without aglets, our laces would fray. Similarly, telomeres protect our genetic information during cell division and prevent chromosomes from adhering to one another. Unfortunately, every time our cells divide, our telomeres get shorter and shorter. This is known as the end replication problem, and occurs because of the way linear DNA replicates in eukaryotes. In summary, the lagging strand contains multiple RNA primers, all of which are replaced with DNA nucleotides, but the one on the extreme 5' end. Luckily for us, we have two secret weapons that help maintain our telomeres, shelterin and telomere terminal transferase, more commonly referred to as telomerase. Shelterin is a complex made of 6 protein subunits that associate with mammalian telomeres. Its function is to help cells distinguish chromosome ends from sites of DNA damage needing repair. To do this, it bends the G-rich 3' overhang so it invades the homologous double-stranded telomere repeat region, completing the formation of a telomeric loop. The absence or disruption of shelterin can lead to telomere doublets and telomere end-to-end fusions, among other abnormalities. What about our other friend telomerase? What is it responsible for? Firstly, human telomerase consists of several components, but the most important are the telomerase reverse transcriptase protein, hTERT, and telomerase RNA, hTERC. Telomerase is recruited during the S phase or synthesis phase of the cell cycle, and its job is to add telomere repeats on the ends of chromosomes. Now, how does it do this? To begin, telomerase binds to the 3' G tail and aligns the tip of a single DNA strand with its RNA template that is complementary to the overhang region. hTERT can then extend the overhang by adding DNA nucleotides that form the telomere repeat sequence. Once the newly synthesized end is created, telomerase can shift to the new end and can repeat this process over and over again. During the next round of DNA replication, DNA polymerase fills in the complementary nucleotides, resulting in double-stranded DNA with additional telomeric repeats. However, unlike germline cells, human somatic cells typically do not express telomerase, and therefore telomere length reduces at each cell division. Why is this a problem? Well, telomere shortening is associated with the development of conditions such as Alzheimer's, bone marrow failure, and diabetes. The rate of telomere loss varies, but is based on cell type as well as genetic conditions like progeria and environmental factors like smoking or obesity. While telomere shortening in somatic cells is one aspect of aging, several other theories also exist. Now, telomeres and aging go hand in hand, but a video on telomeres wouldn't be complete without talking about its relation with cancer, a disease involving the uncontrolled growth of cells, and an area where telomere-based therapies may be the solution. Because cancer cell division is a result of faulty instructions, or a lack of response to said instructions, we need to take a look at the signals involved. One such signal is telomere length. Normally, a cell with critically short telomeres should stop dividing, but immortal cell lines overcome this by activating telomerase or increasing telomerase's expression. Because hTER is mostly absent in normal tissue, but highly active in 85 to 90% of cancer cell lines, an effective anti-telomerase therapy would be a breakthrough in the field of oncology. However, while several ideas related to telomerase inhibition have been proposed, no anti-telomerase therapies are presently approved for clinical use. Several challenges also exist when considering a telomerase-based approach to cancer. A small portion of cancer cells don't use telomeres at all, and instead rely on DNA recombination through an alternate lengthening pathway. However, current developments in telomerase-based anti-cancer therapeutics are up and coming, with promising research in the realm of antisense oligonucleotides and immunotherapeutics. Imetelstat is an oligonucleotide and is one of the most promising anti-telomerase compounds, with testing already being done in clinical trials. Unlike other antisense oligonucleotides, imetelstat does not bind to RNA to inactivate it, but rather binds to a region of the H-TERK sequence that has high affinity and specificity at the active site of telomerase, blocking its recruitment to telomeres. In the world of immunotherapeutics, the primary goal is to sensitize immune cells to tumor cells. Because tumor cells expressing H-TERK peptides as surface antigens can be targeted by cytotoxic cells, several H-TERK and dendritic cell-based vaccines are currently being developed and evaluated. In addition, many novel small molecule inhibitors that target shelterin or telomerase components themselves are also in the works. This can mean inhibiting the tail patch on shelterin protein TPP1 to prevent telomerase recruitment, inhibiting specific alternate splices of H-TERT that can produce catalytically active telomerase, or inhibiting kinases that recruit telomerase, like ATM and ATR. So, have we found the secret behind immortality or cured cancer yet? Unfortunately, no. Telomere length, while a potential biomarker, is only a small slice of the complex topic of aging and cancer, and it's not necessarily the direct cause itself. However, research in the area of telomeres and telomerase may just be the next step for living longer, preventing age-related diseases, and understanding our journey through this strange thing we call life.